Hello friends, Ace Botane, next episode. In this episode, what we'll be discussing is molecular base of inheritance, part 4. So what all things we'll be discussing, for that I will share the screen so it will be convenient for you. I think screen came, so we can proceed. See, in the fourth part of molecular base of inheritance, we'll be discussing an introduction, DNA replication, experimental proof, as well as machinery and enzyme. Then finally, as usual, 5 theory question, 5 NEET model question with a key. So we can enter into introduction. So initial introduction is concerned with Watson and Crick. While proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. The original statement they given was, it is NCERT linked statement. That is why I put there, can be a question. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material, Watson and Crick, 1953. And the photograph of Watson and Crick now you become familiar. Nobel Prize they got in 1962. And the statement is NCERT page number 88, reprint edition, March 24. Now we'll be entering into DNA replication. See, for DNA replication, different models has been suggested, of which finally 3 models become famous. Which are they or what are they? One is conservative, other is semi-conservative, third is dispersive type. 3 types. So what is meant by these 3 terms? First is conservative. Conserve, that is here parental DNA remains together and newly formed daughter strands also together. That is by conserving, conservative. Second is semi-conservative. Here you can find semi-conservative. Here new DNA form. One strand is old and the other strand is new. This is the most acceptable view of the structure of DNA replication. Third, more theoretical method is dispersive. DNA replication produces DNA double helices with part new DNA and part original DNA. Now it differs from semi-conservative, where in semi-conservative, one strand will be completely old, other strand will be completely new. But in third method, dispersive, here parts of old and new together, mixed up like a dispersion. Hence the name dispersive. So most acceptable is semi-conservative model of DNA replication. I think it is clear. Now we'll be proceeding to experimental part. Any experiment, organism, chemical, scientist, purpose. That we have to notice. Questions will be there for theory and entrance. So 1958. Organism used was Escherichia coli. Chemicals, normal nitrogen N14 and heavy isotope of nitrogen N15, but it is not radioactive. Method was centrifugation in a cesium chloride density gradient system. So this part I think clear. Each experiment what all needed. Scientists Matthew Messelson and Franklin Stahl. First page I think it is clear. We can continue with the experiment. See, there is a chemical called ethidium bromide, which is commonly used to detect nucleic acid in molecular biology laboratories. Now, Taylor et al. In 1958 proved the semi-conservative mode of replication by using radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome, performed on Visia fabarum, fababine, Visia faba, it's NCERT. This experiment proved that the DNA in chromosome also replicates semi-conservatively. Later, this type of experiment also done in higher organisms such as plants and human cells. NCERT, page number 88-89, reprint edition, March 24. Because this is all NCERT. NCERT we cannot live. We cannot live NCERT statements. Now, in bacteria, again, called Escherichia coli, a scientist namely J. Keynes, photograph you can find, proved semi-conservative mode of replication by using tritiated thymidine, H3-TDR. In auto-radiography experiment, he also proved theta model of replication on DNA, which is circular. Now, Messelson and Stahl's experiment, photograph given here. Organism, everything now clear. Now we can proceed. Experiment diagram is NCERT, page number 18. It's an important one. Theory question, problem type question, NEET model question. So initially they took the organism with the N15 DNA. That is by red color. Then it is allowed to replicate in N14 medium for 20 minutes. 20 minutes important generation time. So first generation they got where these two DNA strands will unwind and the new nitrogen is N14. So one old strand N15 will be retained. Here also one N15 will be retained, into which new N14s will be added. I think procedure got, because these two will unwind completely. Complete means this part. And N14s will be added. So it become intermediate. One strand N14, one strand N15. I think that idea clear. Then after 20 more minutes, that is second generation. So total time will be 40. So 20-minute generation time. So after 40 minutes, that is second generation, we can find. These two will unwind. And each case, N14 will be added. So this green N14 having another N14, so it is light. This red will get a green, so it will be intermediate. Same with this red will get a green, intermediate. Again here, two light. Simple experiment, but problem type question they can ask. After fourth generation, how many will be completely light, completely heavy, or intermediate? What is the percentage this way? Usual question, easy. And they're done in the experimental side this way. So first case, both strands will be N15. So it will be lying at the bottom of the test tube. Density gradient separation. Second case, it is intermediate, so that will be coming towards the middle of the test tube. And third one, you can find intermediate is here and complete light is here, coming towards the open part of the test tube. So it can be easily elucidated. DNA replication is semi-conservative. So here it is hybrid, that is why middle. Here it is light. Here it is again hybrid. Third test tube, we cannot find N15 and 15. That is due to fact that DNA replicates in a semi-conservative mode. I think it is clear. Important experiment, organism, chemical, etc. Now we can proceed to next page. So a little bit important events will be coming in this episode. Now DNA replication. We have to proceed with the machinery and enzyme, mechanism of DNA replication. So initially I wrote so many chemicals and enzyme involved in this DNA replication with our syllabus. Too many items are needed. It is originally very exhaustive topic, DNA replication, transcription, translation, all this. But what we basically need is NCERT and little slightly above it, that's all. Too much outside, I think no much advantage will be there. Referring to previous question papers of NCERT and NEET exam and theory. So some basic enzymes and chemicals involved in DNA replication. First is helicases. Helical. That is for unwinding DNA. Double helical parallel DNA will be unwinded. It is ATP dependent. Everything I wrote there will be easy for you. Second is topoisomerase. Topoisomerase to nullify the tension released during unwinding. And that works in eukaryote. DNA gyrase, here you can find DNA gyrase to nullify the tension released during unwinding in prokaryotes. Next is single-stranded binding protein, SSBP. What is the purpose? When DNA unwinds, single strand of DNA will not be stable. So it has to stay stable, otherwise purpose will be lost. So stabilizing unwound DNA, it is by SSBP. Next is ligases. Enzyme nomenclature you studied, ligating, uniting. Joins two molecules together here, discontinuously synthesize fragments of DNA or Kasaki. That will be learning later. Sixth is primases. Synthesize or a short primer strand of RNA. And it serves as a stepping stone to start errorless replication. On this RNA only, DNA can come and develop. Okay, so initiation connected with the RNA primer. For that, primase enzyme is needed. Next is DNA polymerase in prokaryote. 3 will be there. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Short form is DNAP1, DNAP2, DNAP3. And this DNAP1, it is called Kornberg enzyme, Arthur Kornberg. That we'll be learning later what is the importance of it. And this is a scientist, got a Nobel Prize in 1959. So DNA polymerase 1, also called Kornberg enzyme discovered by Arthur Kornberg. In 1956, in E. coli, first known DNA polymerase. That is why DNA polymerase 1, like coenzyme 1, coenzyme 2, etc. Main function is removal of RNA primer. DNA polymerase 2 involved in DNA repair. DNA polymerase 3, that is the main polymerizing enzyme, fastest polymerization rate is 2000 base pair per second. That will be studying later. Still not over. DNA polymerase in eukaryote. 5 types will be there: alpha, beta, gamma, delta, epsilon. But that part is not directly in NCERT syllabus. Then 9. Deoxy, ribo, nucleoside triphosphate. 2 functions has to perform. It's important one. One, it is the substrate itself. Second, provide energy for polymerization. So 2 important functions for deoxyribonucleoside triphosphate. 10. DNA-dependent DNA polymerase. That is the main enzyme, highly efficient, fast, and accurate. Now, a statement connected with NCERT that may cause some confusion in NCERT itself, because 2 timing given, 38 and 18, how it came, I'll explain. See, E. coli replication time needed is 38 minutes. That means whole genome of E. coli with the 4.6 into 10 raised to 6 base pairs replicated in 38 minutes. But in NCERT, there is a statement, E. coli completes the process of replication within 18 to 20 minutes. Here you can find, I wrote there, 18 to 20 minutes. So how that 38 and 18 comes? See, what they mentioned, 18 or 20. 20 minutes you'll be remembering. Generation time I told you. Basically, it is generation time, E. coli cell divides. But replication of chromosome needs approximately 48 minutes. That is coming around 38 minutes. That is how this calculation goes. And in a second, we can find 2017 will be checked. That is approximately 2000 base pairs. This is how the calculation comes. That is how the value 18 and 38 or 20 and 40. This is the explanation goes. Otherwise, when we read the NCERT, 2 values given, 38 not directly given, but in calculation side, when we do, it must be 30. Then only you'll be getting 2000, approximately 2000 base pair per second. Otherwise, we'll not get it. This is how that confusion solved. I think idea clear. Now we can proceed to next aspect, the big one, this chapter. So when we proceed to next one, so like ATP, the 2-terminal phosphate in deoxynucleoside triphosphate are high energy phosphates. Whole prokaryotic DNA act as a single replicating unit called replicon, while a eukaryotic DNA molecule has several replicons. Now, energetically, replication is a very expensive process. 12th one, phosphorylase, involved in activating 4 types of deoxyribonucleotides. 13th, energy in different forms, including ATP and GTP. So minimum these all things are needed. NCERT 11. Now steps in DNA replication. Theory entrance question again. We can find 5 steps basically: origin of replication, activation of deoxyribonucleotides, unwinding of DNA helix, formation of primer strand, elongation of new strand. Now first is origin of replication. See, remember, DNA replication, entire DNA will not be involved. Replication begins at a particular region of DNA. That point is called origin of replication, otherwise called ORI. ORI usually rich in AT because 2 hydrogen bonds only involved. So AT base pairs to facilitate unwinding. ORI act as a initiation site for the replication of DNA. Connected with NCERT, again, there is a famous statement. ORI is connected with the requirement of the original replication that a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. Vector here means genetic engineering site. Vector usually provides the origin of replication. Now, prokaryote, there will be single ORI important. Eukaryote, there will be many ORI. And in E. coli, it is refers to be ORI-C. These 3 points important. Second aspect is activation of deoxyribonucleotides. So first we should know what is the need for activation. Because that heading is given and we should know why it is needed. So activation, that is for converting deoxyribonucleotides from their monophosphate form to their triphosphate form. which is very, very important for DNA replication. That is why activation needed. So which is the monophosphate form, which is the triphosphate form, here we can find everything given there. So monophosphate form, DAMP, adenosine monophosphate. Triphosphate, you can find DATP, adenosine triphosphate this way. And for this conversion, energy needed, phosphate needed, enzyme called phosphorylase also needed. I think this idea is clear. Now the third point is unwinding of helix. So for unwinding process, main enzyme is helicases, often refers to be unwindase, is involved, causing separation of DNA strands. That is why the enzyme function I given in the beginning. Now we can proceed to the next page. NCERT diagrams. Comparatively easy. And remember, this diagram is a labeling question, problem type question, and they'll be asking lagging strand, leading strand, what is the direction, all this. So we can proceed. So enzyme need, that is unwindase. For unwinding, ATP is needed. Unwinding will produce a Y-shaped structure. Here we can find Y-shaped structure. And that point is called replication form. Naturally, single strands of DNA will be unstable. So it is stabilized by SSBP, single strand binding protein, or DNA binding protein, DNP. And their DNA separation needs huge amount of energy. That is why from a point it starts. And this is a labeling question, remember. And unwinding takes place in this manner, from here to top. Direction, etc. Check this important diagram, theory entrance, labeling question. Now we can proceed. While unwinding, tension will be created on strands. So it is nullified by topoisomerase in eukaryote, DNA gyrase in prokaryote. Fourth step is formation of primer strand. I discussed what is the need for primer, which is the enzyme, what is the role of RNA. Anyway, we can proceed. So primer normally means preparation, preparatory, or starter, or initial, or stepping. So after unwinding of DNA, respectively, new DNA strands has to be synthesized. In E. coli, DNA polymerase 3, which is refers to be true replicase. True replicase DNA polymerase 3, DNAP3, cannot initiate DNA synthesis. So it is unable to start the synthesis. Means it is not able to deposit first nucleotide in daughter strand. For this, there is an enzyme called primase that will synthesize a short RNA primer strand. So primase, RNA primer strand. So primase is connected with the RNA. After getting that RNA primer strand, on this DNA will be added. Then only errorless replication can proceed. I think idea clear. Importance of primase and RNA primer, then DNA will be added. We can proceed to next page. Later, that means after initiation of DNA synthesis is over, primer RNA strand is removed by DNA polymerase 1, function of DNAP1, Kornberg enzyme. RNA synthesis is involved in the initiation replication of DNA. It has a role, RNA. Next is elongation of new DNA strand. So here DNA has to be added. Enzyme involved is DNA has to be added. DNA-dependent DNA polymerase. But the problem for this enzyme, it can polymerize only in one direction, and the direction is 5' to 3'. Since two strands are there, complication will be there based on direction. Because one strand will be in 3', 5', other will be in 5', 3'. If you check the diagram, it will be clear. So this is an issue in molecular biology, genetics at that time, a complicated issue for a long time. It is solved by a Japanese scientist, Okazaki. Name is Okazaki. Solve this issue. How he made that the problem to solve. Of maintaining the direction for enzyme, suggesting the replication is continuous. See, this is continuous on one template strand with the polarity 3' to 5'. So here 5' to 3'. So new strand will be 5' to 3', anti-parallel continuously formed. There are no problems. And that is called leading daughter strand. So at the same time, other strand which lies in 3' to 5'. Parent strand, here we can find. So replication will not be continuous because part by which DNA can be synthesized to maintain 5' to 3'. So it will be discontinuously synthesized. And short fragments will be synthesized. These short fragments are called Okazaki fragments on the second template strand with the polarity 5' to 3'. And that strand is called, with Okazaki fragments, they are called lagging strand. So leading strand, lagging strand. 5' to 3', 3' to 5'. Here, 5' to 3', 5' to 3'. So all direction can be maintained, can be managed with this idea connected with Okazaki. In prokaryotes, Okazaki fragments contain 1000 to 2000 nucleotides. Not directly NCERT, but if you know, it is good. In eukaryote, these fragments are much shorter, only 1/10 of it, first one, that is 100 to 200 nucleotides. Every time when Okazaki fragments has to be formed, then RNA primer is needed. So here, only one Okazaki means RNA primer is needed. When we come to Okazaki, here one needed, here one needed, here one needed. So more Okazaki means RNA primers are needed in lagging strand. Later, these Okazaki fragments all should be joined together. That is done by the enzyme ligase. Ligase uniting. DNA replication is semi-conservative and semi-discontinuous. Important question. One strand will be old and one will be discontinuously produced. That way. So semi-conservative and semi-discontinuous. Then DNA replication takes place in S phase. Already studied in cell cycle. So S phase of cell cycle. So these procedures should be highly coordinated or can lead to abnormalities, including polyploidy later mutation. After that, 5 theory question and 5 NEET model question with the E. So what all things we learn, we can just pass through. So molecular base of inheritance, that is fourth one, introduction. Then Watson Crick statement, then replication, 3 models, finally semi-conservative. Then experimental proof by Matthew Messelson and Franklin Stoll. Then after that, chemicals, dyes, organism. Then tritiated thymidine, Keynes theta model. Then the experimental part, 20 minutes each generation. Finally here, light, intermediate, AV not there. Easy experiment. Then we entered into machinery, a lot of enzyme, almost 10 or 11. Then I explained how that 18 and 38 comes. So 18 means it is basically generation time. While replication of chromosome need approximately 40 minutes. That is what that 38 comes. And here this calculation, 2000 base pair per second. Then we entered into steps. Origin of replication, ORI, prokaryote, eukaryote. Then by activation needed. Then that famous diagram for labeling. Leading lagging strand, then formation of primer strand, primase. Then elongation of new strand, that is ligases. Then DNA replication is semi-conservative and semi-discontinuous. DNA replication takes place in S phase of cell cycle. Then 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. We can meet later. Thank you.